homologous chromosomes. This is another example of homologous chromosome, in which you can see that these two chromosomes, they are more or less similar, and they have equal length, and they possess long arms. Then, when the chromosomes, they differ in length and function, then they are known as heterologous chromosomes. All of these will come into, we'll be using them in our subsequent classes. Right, now what is the classification of chromosome? Now, based on the centromeres, where the centromeres are present, we classify the chromosomes as metacentric, subcentric, acrocentric, and telocentric. Now, what are they? See, in this, the centromere is lying in the central position. So, the arms are of equal length. Those chromosomes are known as metacentric. Then what about subcentric? Subcentric, you can see, you can compare from this one, that subcentric, the centromere has started moving towards the proximal end, and it is more near the center of the chromosome, such that it divides the chromosome into a short arm and a long. Right? Then subsequently, we have this acrocentric chromosome, in which, again, if you compare from this, the centromere has moved more towards the proximal end. So this is the centromere, which is more towards the proximal end and divides the chromosome into short arm, very short arm, and a very long arms. Okay, and then if you compare the centrosome now in position, then you can see that the centromere has moved to one end of the chromosome, and only long arms are present. Okay, and such chromosomes are known as telocentric chromosomes. So based on the location of centromere, the chromosomes are divided into metacentric, subcentric, acrocentric, and telocentric. Now, before we go further, we all must see that these chromosomes, they are numbered from 1 to 22 according to their lengths, and these are the autosomes, and the sex chromosomes, they are not numbered. You have XX or XY chromosomes. So now if we say that if we are classifying chromosomes according to the position of the centromere, then metacentric will be where centromere is located at the center such that the two arms are equal. For example, now since we have numbered the chromosome, for example, in the metacentric will form the first, second, third chromosome. Whereas in subcentric, the centromere is slightly away from the center such that the two arms are unequal in length, and in this category, majority of chromosomes form. Whereas we have acrocentric, where the centromere, as you have seen earlier, the centromere moves more close to the proximal end such that it has a very short arm and very long arm, right? And examples of such chromosome, acrocentric chromosomes, are chromosomes from number 13 to 15, that is 13, 14, 15, 21, 22, and Y chromosome also falls under this category. Then we have telocentric chromosome. Telocentric chromosomes are where the centromere has moved to one end of the chromosome and only long arms are present in that. And such chromosomes are rare to be found. Then this was the classification according to the location of the centromere. Now we move on to the classification based on the length of the chromosome. And such classification is the standard classification or the Denver's classification, most commonly followed. Now, in Denver's classification, these chromosomes which we have talked of, they are grouped from A to G. Now, how they are grouped? Group A will consist of chromosome number 1, 2, 3. B will consist of chromosome 4 and 5. C will consist of chromosome from 6 to 12 and X. D will consist of chromosome 13, 14, 15. E will consist of 16, 17, 18. F will consist of 19, 20. And last one, G will consist of chromosome 21, 22 and Y. Right? Accordingly, you can see that these chromosomes, A is metacentric, B is subcentric, C also is subcentric, D is acrocentric, E is subcentric. So from this, you can see that majority of the chromosomes are subcentric. Right? And then chromosome 19, 20, they are either metacentric or subcentric, and 21, 22 and Y are acrocentric. Now, this is the chromosome, or this is the Denver's classification or the standard classification, in which you can see group A consists of these 3 chromosomes, group B consists of 4 and 5 pairs of chromosomes, C will consist of chromosome number from 6 to 22, and X and D will consist of chromosome number 13, 14, 15, whereas E will consist of 16, 17, 18, F consists of 19, 20, and G consists of 21, 22, and Y. Now, the third type of classification followed is according to the banding technique used. Don't put your heads much into it, the procedure of banding of each type, because you just need to know it superficially. Right? What are the examples of it? And means there's also one type of classification existing. So, according to the banding technique used, they fall under G banding, R banding, Q banding, and C bandings. Mind you, PGs, if you are seeing this lecture, then PGs ought to know the procedure of each type of banding. Right? Now, so for G banding, a little bit, what you are supposed to know, you use the GM stain, and these stain the AT pairing region as dark, which will signify that it is a heterochromatic region, and GC pairing region as light, bright, which is the euchromatic region of the chromosome. Right? And this type of classification is most frequently used. So this is the example of G banding. Here you can see the brightly stained area, that is the euchromatic areas of the chromosomes, and this is the darkly stained area, the heterochromatic area of the chromosomes. Then apart from that, you have the R banding. Now, R banding has been taken from this word, the reverse of G banding. Now, whatever is light area in G banding will be dark area in the R banding, and vice versa. So, euchromatic region over here, that is heterochromatic region of the G banding, will euchromatic region in G banding, it was taken the stain as light, right? Here it will appear as dark, and heterochromatic region will appear as bright. Right? This is the example of the R banding. Here you can see the heterochromatic areas, that is the dark areas, and the light areas. Then apart from that, you have the Q banding. Q banding has been used by this word, the Crane's mustard stain being used in this, which is a fluorescent dye, right? This will be specifically used to identify the chromosomal and structural abnormalities in the, you'll get to know what is karyotyping in karyotyping of a person. Now, this is the example of the Q banding. Then the fourth example is that of the C banding. CE banding will stain the heterochromatic region, and as all of you know, the heterochromatic region is present in the primary constriction area, the centromeric area. So it is going to stain up the centromeric area, or for that matter, any heterochromatic areas present in the chromosome. Right, nowadays you use this technique, I just thought that you should be familiar. Now this technique is being used, say about 22 years back it had started. So nowadays it is quite frequently used, the fluorescent in situ hybridization, that is the FISH technique, which is now commonly used these days, right? Over here, what happens is that you take up a DNA probe that is a radioactively labeled DNA, and this probe is going to hybridize or going to bind to the metaphase chromosome of the person, wherever its sequence is more or less similar, right? So that is what is FISH in situ hybridization. Right, now wherever it has hybridized, then you do autoradiography and you localize that particular point. What are the various types of FISH probes used? They are centromeric probes, they are whole chromosome paint probes, and they are multicolor spectral karyotyping probes used. So this is an example of the centromeric probes. You can see this fluorescent radioactive areas in the centromeric region, the centromeric probes, where these probes must have bound to the metaphase chromosomes. And here the whole chromosome is painted by these radioactive labeled probes. And this is the example of the spectral karyotype of a normal female. You can see that these chromosomes are differently colored in this spectral karyotyping of a female. Now, you must be wondering that man is using the word karyotyping, and what is it? So, we will see what is karyotyping. Right. Now, karyotyping is basically, what is the chromosomal makeup of a person, that is known as the karyotype of a person, and whatever procedure you will follow for getting this chromosomal makeup is known as the karyotyping. Right, now this karyotyping is generally done to know the abnormalities in the structure or the number of chromosomes. For obtaining a karyotype of a person, you generally take up rapidly dividing cells. The chromosomes in these cells, they are arrested in metaphase. They are fixed, stained, and photographed. This is in nutshell we are talking. We will see them in detail, right? And finally, you take a photograph of that. You cut it out and arrange these chromosomes according to their lengths in A to G groups, and know the kind of anomaly. So, for example, if you've taken a karyotype and if it is of a Down's syndrome, and in which you know that chromosome number 21 is extra, right? So when you're making a karyotype, you will see that where there are 21 chromosomes, there will be 3, right, instead of 2. Now, so we are going into the procedure of karyotyping. I said in karyotyping, we take rapidly dividing cells. So what are the places from where we can take the rapidly dividing cells? These are nothing but the lymphocytes which are taken from the peripheral blood, or the fibroblasts, or the bone marrow cells, the amniotic fluid cells, or the chorionic villus. Biopsy is done to obtain the rapidly dividing cells. But in general, you take up the lymphocytes for this rapidly, as example of rapidly dividing cell. Now, what is the procedure of obtaining these lymphocytes? You draw a sample of blood, about 5 ml. Right. Now, what is the procedure of withdrawing this blood? You all must have done in your physiology classes. Now, you tie a tourniquet. Tourniquet is nothing like a something like a band. Okay, you tie a tourniquet. You ask the person to clench and open the fist again and again, so that there is venous pooling of blood, right? And ultimately, the person will clench the fist, and the sister is going to, sister or a doctor also can be there, will go draw the blood from the which vein is located here in the cubital fossa, the median cubital vein in general, right? So when withdraws the blood, and what is the precaution one must take? That this area, you can see, can you see this cotton? The nurse must have wiped out this area with the spirit, means cotton soaked in spirit. And then once it dries, they start drawing the sample. Right. Now, what precaution they take while drawing a sample? They will first pull up or withdraw the piston of the syringe a little, just to know whether the needle has entered into the vein or not. If it enters into vein, then there will be blood in this your syringe. Otherwise, there will be just air. So you manipulate the syringe without taking it out. And you simultaneously, you should see that you do not manipulate much, because it's a painful procedure. Okay, so then you withdraw 5 ml of blood. Now this blood which you have withdrawn, so you take 5 ml of blood, you centrifuge it to separate white cells from red cells. So you obtain the white cells suspension. To this, you will add culture medium and fetal calf serum, so that these white cells grow in this culture medium, and fetal calf serum is going to provide nutrition to these 5 white cells. Now, after that, you will add phytohemagglutinin and antibiotic to this. Now, this antibiotic is going to prevent infection of the culture medium, and phytohemagglutinin helps in cell divisions, further cell division. And then this particular thing you will be incubating in at 37 degrees centigrade for 3 days, right? After that, you add to this suspension the colchicine. Colchicine is added so as to arrest the metaphase in the cell division. The cell division is arrested in metaphase. So you get the metaphase of the chromosome. Now, to this suspension, you add hypotonic saline. Now, this hypotonic cell line will make the cells swell, the chromosome separate, and this ultimately you take a drop of it from the pipette and you drop it on the slide from some distance. Why you want to drop it from some distance? Because so that the cells rupture and the metaphase spreads, right? This metaphase spread is fixed up and stained. Then this metaphase spread, this is entirely the structure now on the slide, is known as metaphase spread, which is then photographed, and the chromosomes are cut from the photograph and then they are karyotyped from A to G groups. So this is the chromosomes which have been karyotyped from A to G groups. And then we come on to sex chromatin before we go on to Lyon's hypothesis. Now, just understand, sex chromatin you will observe in interphase part of the cells, that is the resting cell nucleus will be interphase nucleus. Over here you will observe that in the nucleus of the cell there is a condensed mass which is the chromatin in structure. It is present towards the inner aspect of the nuclear membrane in females. Mind you, it is present in females and it is not present in males. This chromatin structure, the condensed structure, you'll get to know why. Now, most of the body cells will possess chromatins, right? Now, these chromatins, they are observed in cells, although they are present in all the cells, but most commonly they will be found in cells derived from skin, buccal mucosa, vaginal epithelium, urethral epithelium, or blood cells. But most commonly used, a non-invasive procedure is from the buccal mucosa, cells from the buccal mucosa. Right now, what are the methods of observing these sex chromatin? How you will obtain these cells, rapidly dividing cells. The cells present in the scraping of the cheek, right? You just take a scraping of the cheek and then you evenly spread it on a slide, fix it in alcohol, then you stain it with basic dye and observe under microscope in high power. Right? What do you observe? That each cell will possess chromatin, sex chromatin, if it is being taken from a female. Males, mind you, will not have the sex chromatin, right? So you observe the buccal mucosa cell. This is just one cell. Of course, you will have many, right? So nucleus of the buccal mucosa cell, and you can see that at one part on the inner aspect of the nuclear membrane is present the condensed chromatin, that is the sex chromatin. Right, sex chromatin is also known as Barr body because it was first demonstrated by Moray L. Barr. Right, and you all know that this sex chromatin is nothing but inactivated X chromosome, which is going to be found in females and not in males. So in females you will have the presence of Barr body, in males you will not have the presence of Barr body. This is the Barr body or the sex chromatin present on the inner aspect of the nuclear membrane, right? And then you have in Klinefelter syndrome, since you have the presence of X, a combination something like females, so this X will be, one of these X will be represented as a Barr body or the sex chromatin. So you will find in Barr body or sex chromatin in females in a person having Klinefelter syndrome? And will you have Barr body in a person having Turner syndrome? No. Why? Because the genetic makeup is X0. So there is no Barr body present in Turner syndrome. Okay. Now, how will you calculate the number of Barr bodies? Remember, the Barr bodies are always one less than the total number of chromosomes, right? So, for example, in males, if the genetic makeup is XY, since you have just one chromosome, so one less of total number of chromosomes, sex chromosomes, will be no Barr body in males. Whereas in females, since you have two X chromosomes, one will represent as the Barr body. So you have one Barr body in females, and in Turner syndrome, as I told you earlier, since you have presence of just one X chromosome, you will have 0 Barr bodies. And in Klinefelter syndrome, since you have going to have the genetic makeup is XXY, so you're going to have 2 X's, and then one of X will be represented as Barr body. Now, which X is inactivated, and why X is inactivated in females only? See, females have 2 X, so the protein, if they form the protein, that there will be double amount of protein in females compared to males who have just 1 X, right? And this double amount of protein is going to be very lethal to females, right? So what happens is that one of the X is made into sex chromatine or inactivated, and so there is just presence of one X in females, one active X in females also. One X is inactive. Okay, so the same amount of proteins will be formed in males, proteins formed off by X, and the same amount will be formed in females. And this inactivation of X chromosome is known as dosage compensation. So this dosage compensation occurs in females. Now, apart from seeing Barr bodies, you can also identify that this particular sample is from the female, or this particular sample has more than 1 X, will be presence of drumsticks. Now, what are drumsticks? Drumsticks are nothing but this type of structure which is present if you take a peripheral smear in the neutrophils. In the neutrophils, you will find this drumstick-like appearance, but generally you may not find in many of the cases. It's a very unsatisfactory procedure of knowing that this particular slide has sex chromatine or not, or if 1X is inactivated or not in this particular slide. Now, what is Lyon's hypothesis? Now, Lyon's hypothesis states that only one of the 2 X chromosomes is functional in females, as we have seen, and the other gets inactivated. So this was hypothesized by Mary F. Lyon. That is why it is known as Lyon's hypothesis, right? And this inactivation of X chromosome, it occurs early in life, right? Early in life, and it can be either inactivation. See, in a particular somatic cell, 1 X chromosome, if it is a female sex, 1 X chromosome will be derived from mother, and 1 X chromosome will be derived from the father. So which one of them will be inactivated, whether it is mother's chromosome or it is father's chromosome in one particular cell, is at random. You do not know. So, as I told you, it was hypothesized by Mary F. Lyon in 1962, and this occurs basically at a very early stage of development, that is at a 15th to 16th day of the development, and one of the two chromosomes, they are inactivated in the form of and represent in the form of Barr bodies or sex chromatine, and this process is known as Leonization. Now, what are the features of Leonization? So we are just summarizing now. They are that this Leonization, it occurs early in embryogenesis, where one of the two chromosomes is inactivated. Which chromosome is to be inactivated? It is chosen at random. It can be either maternal or paternal in a particular cell. Then, if suppose mother's chromosome is inactivated, X chromosome is inactivated in particular somatic cell, then after it divides and redivides, it will always follow that mother's chromosome is only inactivated, not the father's. So, another example, if suppose in a particular somatic cell, the father's X chromosome is inactivated, then whatever progeny it has after mitosis, then the same father's chromosome is going to be inactivated in the progeny of that particular cell. It is not going to be the mother's. Okay, so this is meant by this particular sentence, that once a particular X chromosome is inactivated, the same will be inactivated in all daughter cells. Right, so we can say that a female is kind of mosaic. Now, what is mosaic? When the individual is composed of cells which have two genetically different types. So some cells are maternal, some cells are paternal derived. Okay, with respect to genes. And mitosis of the inactive X chromosome will occur late when replication is occurring, and that is it for today. And then so we can just a minute. Yeah, so now to summarize, we have done today the chromosomes, their classification, then we have known what is karyotyping, and we also learned about Lyon's hypothesis. We learned about sex chromatins and sex chromatin, also known as Barr bodies. Right. So this is all for today, and now you people can ask me questions if you have any. You can just type in your questions.